kind of want to preface by saying that at any given moment, there is like an absolute actual number of mosquitoes flying around out in Fort Collins or anywhere in the world that are infected by West Nile virus that are capable of transmitting it. And how many of those there are and where they are and to what degree they interact with other people determines how risky a situation is. And so the goal of our study, the goal of our work with surveillance is to try to provide some information on what that risk is, what's the distribution of risk in space and time, and to provide some data that can underpin efforts to limit the impact of West Nile virus or any other virus to people. And what that can look like is really different. There's all kinds of ways that you can do that. But having some actionable data on how risky, how dangerous is a situation is generally useful. And so that's what we're trying to do. Okay. So I'm going to attempt to share these slides. Are you looking at this thing that my cursor's on right now? Yeah, okay. So this is a talk that I first gave back in February, and so I'm going to use some of this, and then we'll probably diverge. And please, if you have questions, like don't just speak up. Let me make sure that I have my speakers turned on. Actually, I don't know how I can do that and still keep sharing my screen, so I won't. Can someone just say, well, Tim, you said something already, so I already know that. Okay. So, all right, since this is maybe the second talk that you've had, let's keep it general. Again, if there are questions, ask them. So what I kind of have for you is background on West Nile virus. I'm going to cover some of the very basic virology. As I said, some of the clinical disease pathogenesis, epi, and then West Nile surveillance in Fort Collins. And then this background, this is stuff that was for a class and they were doing a serological study. And so I wanted to give them some background on that. But we won't go into that unless you tell me that you're super interested in it, and then I will. Okay, so just by way of starting, I think that it's important to recognize that arthropod-borne diseases are have this really unique capacity to cause explosive outbreaks. You can have entire communities kind of infected in what is basically overnight. You're part of the center, so I probably don't have to convince you that in general, arthropod-borne diseases are some of the most persistent and burdensome health problems that we have to confront as people. And we're not just talking West Nile virus, we're talking malaria, lymphatic filariasis, several arboviruses, and many of these are emerging and re-emerging infections that are emerging and re-emerging due to very kind of larger, very complicated problems that we as a human race on planet Earth have. One of the reasons that they're so persistent and burdensome is that they persist in these really complicated transmission cycles. If you ever kind of sit back and think, why would a virus pick to be arthropod-borne? It's kind of makes you wonder, right? Like, if you could just focus on a vertebrate T cell or something, that might be a little bit easier than having to do this weird thing where you go back and forth from a mosquito that's flying around. It's going to get rained on, or maybe it'll get too hot and dry and it's going to drop dead, and then somehow get into another animal, and hopefully that animal's not immune already, or it has the right receptors for your virus. So there's all kinds of ways that cannot work. And so the complexity of the transmission cycle is always something that to me has been kind of interesting. Mostly, I think it's quite accurate to think of arthropod-borne disease as environmental disease. These are pathogens and transmission cycles that really emerge due to the human footprint on the environment. And that's true from malaria right on down to West Nile virus. So I'm going to spend a few minutes just talking about the virology of West Nile virus. It's part of a really large diverse taxonomic grouping of viruses that include mosquito-borne viruses. There's a few names of them here. West Nile is part of this Japanese encephalitis virus serological complex, but there's a lot of these other things that you've never heard of. Here are the dengue viruses. Here's yellow fever viruses. That's the clade that's mosquito-borne. And then there are also a big group of not mosquito-borne, but tick-borne flaviviruses. And then some that have no known vectors. And then there's this group of arthropod-specific viruses. This is pretty interesting, and we're not going to talk about it today, but there are actually other arthropod-specific viruses that are interspersed over here with the mosquito and tick-borne groups. And so that's just kind of an interesting evolutionary conundrum, I think. Worth knowing, although not germane to what we're talking about today, is that the flavivirus genus that I'm showing you here is related to several other important viruses that are member of the same virus family, which is Flaviviridae, including hepatitis C virus and then some animal viruses over here that cause diarrhea in animals. I think that's all I want to really say about that. The virus itself is really genetically and phenotypically diverse. And so there are these two kind of like well-defined West Nile viruses lineages. Lineage 1 and lineage 2 both originated someplace in Africa. They also both seem to have distinct pathogenic potential. Lineage 1 is the virus that has the New York strain here. This is the virus that we have in the US. This tends to cause encephalitic disease, whereas lineage 2 does not. Yeah, we focus on lineage 1 because of its importance in our area. Also important to say that classification of viruses is always changing, and it's pretty annoying. I try to stay out of it, but yeah. Okay, so West Nile virus is a flavivirus. It's in the family Flaviviridae. It's a single-stranded plus sense enveloped RNA virus. And so what that means is that its genome is made of RNA, and every virus particle like this has one strand of RNA in it, and that's the virus's genome. That's all that it is. There's no DNA there. There's no viral translation machinery. It's just a strand of RNA viruses, and it's plus sense. And so what that means is that when it's positive sense RNA, that means that the viral RNA that comes in the virus particle is able to immediately make protein, and then that protein can start making more virus RNA, which can then make more protein. So if it was negative sense RNA, well, what that would mean is that the virus would have to package some form of its replication machinery to take the negative sense RNA and make it into something that the cell could read, which would be the positive sense RNA. So that basically when we say that this RNA is infectious, that's what we mean. We could take this RNA outside of all these things and electroporate it into a cell, and it would start making viral proteins. Are you all with me so far? Okay, cool. This is the genome of West Nile, or really any flavivirus, and most Flaviviridae have motifs that are quite similar to this. There are only 3 structural coding sequences that makes 3 structural proteins: the capsid, which binds RNA, the PRM, and the E. And the PRM is a pre-membrane, and then the E stands for envelope, C stands for capsid. And these things are present on the surface of the virus particle. And the E is responsible for virus attachment and entry and getting the virus stuck onto the cell and getting the genome into the cell. And the pre-membrane and membrane proteins are responsible for shielding certain portions of this envelope protein as the virus is moving through the cell and getting out of the cell. And then there's these 7 non-structural proteins down here. And so what these things are doing is replicating the viral RNA and basically antagonizing the host antivirus response. So the cool thing about these viruses, in my opinion, is that all these proteins do lots and lots of different things. All of the proteins are multifunctional. And so, NS1, for example, is secreted and it can poke holes in membranes. In addition, it serves as part of the scaffold that the RNA replication complex sits on in cells. So pretty cool, multifunction protein stuff that goes on with these viruses. It's like a really efficient, compact set of stuff that it does. Yeah, generalized flavivirus life cycle. I think it's important just for you to kind of see what happens when the virus gets in the cell, is that it sticks onto the cell somehow. We don't really know how. It gets into the cell and it's through a process of endocytosis. And an endocytic vesicle, as it comes into the cell, starts to acidify. And as that packet acidifies, it causes the envelope proteins on the surface of the virus to undergo this pretty dramatic conformational change that ultimately results in the viral genome getting stuck into the cell and starting to replicate. RNA replication happens in these really specialized factories on host cell membranes, and then viruses bud out through the Golgi apparatus that you learned about back in high school. And I said this part to you already. All right. So now let's kind of move on to kind of more of the epi and the clinical parts of all of this, also, which I think are important. I mean, a lot of what we do in RaHP VEC is around West Nile virus, and I think it's important for you to kind of know why this is a problem for us. So this is from last summer, and way more than 11 people ended up dying from West Nile in Colorado last year. It was like literally the worst West Nile year that I've known since I came to Colorado back in 2011. It's just a news item. But this is not from Colorado. This is from West Nile virus since it's been in North America. We have a lot of huge number of cases, a lot of hospitalizations, and nearly 3,000 deaths have occurred so far. So just to kind of put those numbers in perspective, West Nile is now, without a doubt, by far the most common cause of arbovirus encephalitis in the Western Hemisphere. There's an estimate based on how frequently people get sick compared to how frequently they're infected. It's estimated that about 3 million people have gotten infected by West Nile virus since it was introduced into North America, 24 years ago now. But that means that there's still 328 million people out there to infect. So we're not even close to having enough people, be infected that there's actual herd immunity to it. But what we think is likely to happen, and this has been seen over and over again, is that the epi is going to be characterized by outbreaks that happen in the summer when the mosquitoes are flying around. And they will be more or less severe, and we've had a few very severe years so far. Okay. So if you think about West Nile virus, there's a sort of this iceberg idea. So it's thought that about 80% of individuals that are infected by West Nile virus remain asymptomatic. Of the remaining 20%, the majority of them get something that's called West Nile fever, which from what I understand, I've never had West Nile myself, really doesn't feel very good, but you sort of, it's what's called a self-limiting febrile illness. You get a fever, you feel bad, and then in a few days later, you start to feel better, and then ultimately you're fine. Less than 1% have what's called like West Nile virus neuroinvasive disease, which is what happens when the virus invades the central nervous system and starts to cause damage. And the way that it causes damage either is by directly injuring the cells, so the virus infects the cells and causes them to die, or it infects the cells and the cells are, while not happy, not going to die, but then your immune system comes in and causes significant inflammation in the brain, and that can cause sort of ancillary auxiliary damage to CNS. All right, I talked to you about this already. I talked to you about that already. Yeah, so inflammation in the brain is meningitis, encephalitis, poliomyelitis-like syndrome. That's very, very bad, very sad. And then also I didn't mention necrosis, which would be due to direct injury by the viruses. The acute disease is characterized by increased age. Immunosuppression are both independent risk factors for neuroinvasion. The acute case fatality in West Nile virus neuroinvasive disease is about 15 or 20%. So if you are one of the unlucky 1% that get neuroinvasive disease, then you've got about a 15 or 20% chance of dying. There's been this really sort of classical problem with survivors of West Nile virus, and they have this kind of like movement disorders. And so you ask somebody to stand up or to move or to hold their hands steady, and they have no ability really to do that anymore. So that's sort of indicative of long-term neurological damage. And then there's this poliomyelitis-like syndrome that happens occasionally with West Nile virus. It usually happens in younger kids, and it's quite severe. It has a very poor long-term prognosis. The other thing to think about is not just that there's acute disease due to West Nile virus, but there's these very long-term effects that can be really problematic. Persistent symptoms have been common. There was a study in Chicago back in the 2000s that showed that 63% of just West Nile fever, right? So these aren't neuroinvasive disease, had persistent symptoms that lasted for more than 30 days, and in some cases lasted 60 days. It can really sort of impact people's quality of life over time. And that's even just for West Nile fever. West Nile encephalitis, you can really have these like persistent disabling neurological sequelae. You can really think about it as like brain damage that you have because of West Nile virus infection. I see there's something in the chat, so I want to make sure that. Okay, that's Tim. All right. So I talked a little bit about West Nile poliomyelitis. It's very high acute mortality. You sort of think about polio back in the early part of the last century and people getting stuck in these iron lungs, and that has happened with some of these West Nile cases. Long-term weakness is really severe, and this often is very, very much kind of younger people. So that's a really, really particularly tragic outcome of West Nile. Fairly uncommon, but unfortunate nonetheless. So, all right, just to summarize on the clinical disease, West Nile is associated with substantial morbidity and mortality, particularly in high-risk groups, which can be very young and very old people or people with other immune suppressions. Overall case fatality rates, and those are people who show up at the hospital or who get sick enough to have to go to the doctor, about 1%. We're understanding increasingly now that West Nile and other arboviral encephalitides are associated with long-term problems. Loss of neurons, renal damage, cortical thinning, and early mortality. And this has been shown over and over. Okay, so West Nile virus, as you likely know, is maintained in nature in an enzootic cycle between mosquitoes, principally of the genus Culex, and ornithophilic birds, and passerine birds. Ornithophilic mosquitoes and passerine birds, sorry about that. Excess avian mortality and disease in horses and humans occurs by a spillover from this main enzootic cycle. Of course, there's more to it than that. So we can sort of think of this regular enzootic cycle that goes back and forth between mosquitoes and birds, but there's also very good evidence that fecal-oral transmission can occur in birds. So there's a very good example of a crow roost that had a massive mortality due to West Nile virus in the middle of the winter. It was like in February in upstate New York, and there was no way there was any mosquitoes around. So the idea is that the bird somehow became infected and then died or was defecating, and then the other birds were feeding on the carrion, and just this whole roost of crows got wiped out. It may be possible that there are other vectors that are involved. It's been shown that ticks can become infected, although that seems to be very, very unlikely or very uncommon. Mosquitoes are able to transmit vertically. And then, as I mentioned earlier, you have this epizootic, so you have excess avian mortality, infection in animals not typically seen infected, and then we don't really know the degree to which any of these other animals have infectious viremia, but it's thought to be fairly uncommon that they would have enough viremia to contribute to transmission. By far, the most important transmission cycle is here between mosquitoes and birds. So I'm going to skip through this because I think I want to spend some time talking about this next concept, which is vector competence. So has anybody talked with you about vector competence in this context yet? No, okay. So this is like really a key concept. And so what people used to think is that all mosquitoes were able to get infected by a given arbovirus. So like any mosquito that fed on a bird that had West Nile would get West Nile. What we know now is that that's definitely like not the case. So there's species of mosquitoes that are extremely good vectors and mosquitoes species of mosquitoes that are extremely bad vectors, given exactly the same infectious dose that they feed upon. So the way that viruses work in mosquitoes is that they're acquired when the mosquito takes a blood meal. That virus then has to get into the mosquito's midgut, attach to the midgut epithelial cells, and then replicate within the midgut. After it does that, it has to escape the midgut into these outside tissues. And so mosquitoes don't have like a circulatory system like we have have what's essentially an open circulatory system. And so all of their tissues are bathed in this fluid that's called hemolymph that also has various immune and blood cells within it, but it's not like pumped through veins like ours is. After the virus escapes from the midgut, it then sort of is in this hemolymph and ultimately infects the salivary glands, which is this green thing that I'm trying to point at here on the pointer. So those are the mosquito salivary glands, and it has to do the same thing there. It has to infect the mosquito salivary glands, except this time it's doing it from the outside. And then it has to replicate within those cells and then get released into the lumen of the mosquito salivary gland. And then when the mosquito feeds again, it can get ejected into the skin of whatever it's feeding on. So when a mosquito is biting you, it's like spitting the whole time, which is why you get a mosquito bite that's itchy, because you have an allergic reaction against the proteins that the mosquito is feeding in there. So this is this panel B thing is actual data on different mosquito species that we have in the lab out here at CVID: Culex tarsalis, Culex clinicofasciatus, Culex pipiens, and Aedes aegypti. And what you can see from this is that, first of all, hopefully your eye tells you that Culex tarsalis are more competent than these others. They almost all are infected. They almost all have infectious virus in their legs, in their salivary glands, and that most of them that have virus in their salivary glands are transmitting the virus. Culex fasciatus and pipiens also are very susceptible because they have virus in their midguts, but they have stronger barriers to escape from the midgut or infecting the salivary glands or getting out of the salivary glands and into the saliva. And so that's why you see this kind of dropping down. And then Aedes aegypti are more resistant to infection. They're less susceptible, but their physiological barriers are less. So you have this kind of interesting situation where Culex and pipiens and Aedes aegypti are all about the same amount competent, but they're the same amount competent for really different reasons. I think the take home for this for all of you is that Culex tarsalis are just like hyper efficient vectors. Similarly, the different species of birds might have different, competence, ability to support virus replication and give mosquitoes a infectious amount of virus in their blood. And so this just is a summary slide. It's just to make the point that, in addition to mosquitoes being different in their competence, vertebrates also can be different in their competence. Vectorial capacity also is a key concept that I want to introduce you to because I think it's important. Like if you can remember vector competence and vectorial capacity, you're going to be like 50% of the way towards knowing most of what people think they know about the transmission of vector-borne diseases. So vectorial capacity is trying to get at something that's a little bit different, which is not just like, is this a good vector or not? But like, is this a good vector that also is interacting with the right kind of animals and is living long enough and is getting the virus ready to be transmitted fast enough? So this is the equation. Vectorial capacity is basically like how many new infections occur from a single infection that's given into a given environment. So the terms I'm just going to walk through them. Please don't be scared. It's math, but it's really not that bad. It's M is like how many mosquitoes are there. A is the degree of host focus, because a mosquito has to feed twice on any given animal in order to be able to be any kind of a vector, right? So if this is West Nile and it feeds on a bird, and then it feeds on a horse, well, the horse might get infected, but it's not contributing to transmission. It's not contributing to the perpetuation of the agent in nature. P is the probability of daily survival. So life is dangerous for mosquitoes. And if the mosquito gets infected and then it dies 2 days later, well, that doesn't do any good because it hasn't lived long enough for the virus progress all the way through all the different tissues that it has to get through in order to be transmissible. So P, as I said, is the probability of daily survival, and N is the extrinsic incubation period. So the number of days that it takes for a pathogen, once it gets ingested by a mosquito, to be transmitted again. And then this is vector competence. So what hopefully this can tell you is that things like the degree of the host focus and what that mosquito is feeding on, and then how likely it is to survive each day are like really, really important in determining how good or bad of a vector it is, besides just the biological compatibility of a virus and a mosquito. Does that make sense? All right. If I could pick two things for you to learn, it's vector competence, what is that, and what is vectorial capacity? And you don't have to even know the math, just say that it incorporates all the other kind of ecological stuff. All right. Okay, so surveillance. I get to it. So surveillance can sample at different points in the West Nile virus system. So we sample people. We learn about how bad the virus is by looking at people. That West Nile virus is a reportable, notifiable disease, and so that's one way that we can learn about it. We can say, okay, who shows up at the hospital and who shows up at the clinic, and do they have West Nile or not? We can also do surveillance in animals. Horses get vaccinated for the most part now, so we don't see too many horses that are clinically ill, but we see lots of birds most summers. And that's another like very sensitive way of detecting West Nile virus is if birds start, either dropping out of roosts or they show up at the Raptor Center, who we collaborate with on West Nile virus surveillance, and they're neurologic, they frequently are infected by West Nile virus. So you can also sample in the environment. So to me, this means mosquitoes, and that's what we do, and hopefully I'll be able to tell you more about that. But you could also imagine sampling wastewater. There's a project that CDC is quite interested in looking at, sampling wastewater for evidence that West Nile virus is present in a given environment. And then you could also just kind of look at how land use is changing and look at what agricultural practices there are and what kind of temperatures and rainfall and snowfall there are, and use these things to kind of try to make predictions. It's not really surveillance, in my opinion, but there are also factors that could play into how you think about the data that you're getting from mosquitoes, wastewater, horses, people, and so forth. The point being, though, that we have this complicated transmission cycle, and there's a lot of things that we can do to try to understand the degree of risk that might be associated with a virus in a given environment at a given time and place. So I said earlier that 2023 was a really bad year for us in Colorado. This shows the cases in 2023, and this is the average of the previous 5 years. We were at week 44 when I pulled this off the website at CDPHE. But we had, like over 46 deaths last year, and just a lot of people got very sick. We don't see this often at all. You can see this is what a typical year is, and we were way, way, way above that in terms of cases.
I have a quick question for you.
Yeah, sure.
So when these big spikes in the human cases come up, how much earlier would you see like a spike in mosquito populations? Would it be like a week earlier?
Well, you'll have to hold this in your mind to see that. But yeah, typically what you see first is the number going way up. And then
Mm-hmm.
second, in those mosquitoes, typically you will start to see virus prevalence come up. Often we'll see birds that are dead before we see any of that, and then we see the people come up. So that's why what we're trying to do is look at the mosquito populations and infection in mosquitoes, because that typically gives you a couple of weeks of warning before you start to see these huge spikes of cases, which in theory is enough time to take some action to suppress the populations of mosquitoes. You can see bird positives really, really early on in the season. So early, in fact, that they tend to be like not that informative. When I was working at the New York State Health Department, we got birds, we would start seeing birds positive at the end of March, but nobody would get sick until the end of it. So we stopped testing birds because it was like, well, they're going to be positive, and we're not going to know anything about like when we're going to go out and do anything. Does that make sense, kind of?
Yeah. And then I guess my other question for that is, do you know which species is contributing the most for like human cases? Because I know like Tarsalis is more of a rural species. So I'm wondering if you know if it's more of like Tarsalis that's doing this,
Yeah,
or if it is like, like in town, if it's maybe one of the other Culex species.
yeah. So most of the human cases kind of happen around the transition when Tarsalis is starting to decline from really early season peaks and pipiens are starting to kind of be a little bit more important. And so, like, of course, we're never going to know which mosquito transmitted any given infection. But the way that I kind of think about it is that Culex tarsalis are probably what's really driving enzootic transmission. And I think they're really effective at doing that and generating just these huge pools of
Like
infected
pools, yeah.
mosquitoes. Yeah. And then, so some of them maybe are, because I mean, I get bit by tarsalis in my backyard. Some of them are probably transmitting in some cases, but you could also get um, the Culex pipiens that are kind of starting to come up later in the season when we see a bit more of our cases. I think it's both. I don't think you could really, I would never say definitively it's all pipiens or it's all tarsalis. I think the truth is that it's a fair amount of both. And it is context dependent, as you say. I mean, we get tarsalis in the town of Fort Collins. So they are rural compared to like urban Culex pipiens. You wouldn't get Culex tarsalis like in New York City.
Mhm.
Um, but we get them, we have them all around Fort Collins.
So it could be a case that it's more that the Tarsalis could just be making sure there's a really nice big transition or a transmission pool ready to go right when those pipiens are popping up too. Like they've already infected all of, I don't know, robins or something, and then they're coming into town or
Yeah,
something like that.
yes, that's kind of how I think about it. I don't know that that's like 100% right, but that's kind of how I kind of think about it. Is that at least Tarsalis are driving that kind of early expansion, and they get the cycle going. And then after that, it's like anybody's guess. It's like just kind of whatever mosquitoes are there. Does that make sense, kind of?
Yeah, that answers it. Thank you.
Okay, just a minute now. I have to find myself again. All right.
Greg, Anna also just asked a quick question in the chat. Just basically, is it possible for like direct transmission from the bird or another host, like host to host without the vector? Figured I'd let you answer that.
Yeah, so that's an interesting question. That's definitely possible. So one of the ways people think raptors get infected sometimes is by eating birds that are themselves infected. Nick Komar showed many years ago that you could, feed a bird a virus-infected mosquito and that they could get infected that way. Um, there's evidence of fecal oral transmission and also evidence of carrion feeding transmission. So there's a lot of ways that you can that a bird anyway could get it. Does that answer your question, Anna? Is that what you were looking for?
Can you hear me?
Yeah.
Okay, I was more wondering if a person would be able to contract it from a bird or a horse, or since, you need the mosquito to get it in the bloodstream, if that's not really a concern.
Um, I mean, I would advise anybody who finds a dead bird to, not interact with it in too intimate of a manner. I don't think there's like a huge risk of like, picking up the bird and disposing of it. But if I was doing that, I would, maybe get a pair of gloves and stuff. The virus isn't effectively transmitted by aerosol, although if you got a high enough amount of virus in there, you probably could make it work if you tried. Um, I want to say that there was some studies very early on of um, infants becoming infected during nursing on moms that were infected, and there's also a transfusion risk. So the blood supply is screened for West Nile virus. So there's those things. Yeah. All right.
I also read a paper recently that suggested that alligators could serve as amplifying hosts for West Nile virus, at least in the American Southeast, and that they could transmit it to each other. Do you know the mechanism from which they transmit to each other? They're like excreting it via like their cloaca or something.
Yeah, that would be it. And we have a paper on that recently. I don't remember the details of it. Early on, alligator farms got whacked by West Nile pretty bad. I don't remember the literature on that, Zane. Do you remember the first author on, I'm embarrassed because I probably was an author on that paper and I don't remember it enough to details on it. But if it was Alex Bias was an author on it, then yeah, I mean, she was able to show that they would get infected, but she didn't ever saw in her experiments a lot of like alligator to alligator transmission. So I think that it can happen. Although if you're in that kind of a situation, the alligator farms are kind of outside, and so they're probably getting bitten by a lot of mosquitoes too.
Yeah, it was the bias paper. I do see you on that list too.
I'm sorry. Alex was
I
a
mean,
great grad student in my lab, and that was like one of her side projects that she did, and Angela Bosco-Louth was the main person that she worked with on that one. Okay, so I'll move on a little. How are we doing on? Yeah, okay, 12:42. We have a little time still. Okay, I'll move on from this. This is some of our data, just again emphasizing, 2023 and how you could conceivably do surveillance with bird samples. We haven't plotted the 2023 data yet, but you can see very clearly that proportional to the humans, 2023 was also a very bad year for the raptors in our study. I wouldn't worry too much about this. We just, we went from 12 positive raptors in 2021, 43 positive in 2022, and 73 in 2023. And if you look at 2021, 2022, and 2023 here, we go like, from 19 in 2020, 113, okay, that's a little bit worse. 2022, 143, okay, that's a little bit worse. And these are just the hospitalized people. And then we've got 300 in 2023. So that also is bad. Okay, so we do quite a lot of surveillance during the summer months for West Nile virus in Fort Collins. And here's a map of where these traps are. And so what we're doing is we've been sampling mosquitoes from these same traps basically for the last 20 plus years. I've been here since 2011. We started working with the surveillance program in 2012, 2013, and we've been doing this since then. This has been kind of an interesting and fun project. And part of the reason that it's interesting and fun is that we get to work with people at the city of Fort Collins and at contractors who do mosquito trapping and identification. We're in contact always with people at county and state health departments. And then we also get to work with the Raptor program and that data. So the way that the surveillance works is that the traps are set weekly by contractors. Will and his group, this is Vector Disease Control International. They set these arrays of traps and they collect them in the morning, collect the mosquitoes. The mosquitoes go back to the VDCI shop and then they are knocked down and identified. After they're identified, they're placed into pools. And so we only test pools of vectors. So we're only testing Culex, mosquitoes, Tarsalis, and pipiens. The pools can go into size like up to 50. And then they get in a tube that looks like this one here. This is a round bottom 2 ml tube with a glass or a steel ball bearing. And then we add buffer, which is a mosquito diluent buffer that has antibiotics and antifungals. And then they're ground up in a mixer mill like this. They basically, each one of these like kind of clampy things, you have like a white seating that you can put like 24 of those tubes in at once, and then it just shakes them up really fast and grinds the mosquitoes kind of into just kind of a mosquito sludge that you then take out and you spin them down so that the sludge parts and the BBL go to the bottom, and then you can draw off the supernatant at the top. And you don't have to grind the mosquitoes that hard. You just have to make it so that their cuticle gets disrupted, and that will express all of the virus that's in there. Any virus that's in there will get into this solution. So then some of that solution out of these tubes that's been then spun down so that you have a clear supernatant is put into our robot, which we have in the lab, and RNA is extracted. Oh, here's a picture of what it looks like. There's the BB and there's the mosquito bits, and then there's the soup. So once we have RNA from this, we then test it by quantitative real-time PCR. Does everybody know what quantitative real-time PCR is? Yeah, okay. It's basically a method, for those of you who don't know, it's a method for detecting nucleic acids. So if I say PCR, that stands for polymerase chain reaction. And what you're doing is you are adding enzymes and short stretches of DNA that recognize a DNA target. And then by changing the temperature, you can change the activity of the enzyme and the structure and state of a DNA molecule. So if you just imagine that DNA is double-stranded and it's stuck together like this, if you heat that up, the two strands come apart. They dissociate from one another. So you have these free strands of DNA. And then what you've added to your mixture is short stretches of DNA that are able to recognize by sequence some piece on this other piece of DNA. And you have two of them that let you go in different directions. So you try to amplify a chunk of the DNA. So you heat the thing up, you heat the DNA up, it dissociates, then you cool it down, and the primers come in and bind to the different places on the DNA strand. And then you heat it back up to like an intermediate temperature, and that's when an enzyme is active that's able to polymerize the DNA. And you just, in that way, by doing that like 40 times, you can amplify the chunk of DNA that you're looking for. So I told you that these are RNA viruses. So what that means is after your extraction, you have not DNA, but you have RNA. And so there's an additional step where you add an enzyme that's called reverse transcriptase that takes the strand of RNA and turns it into DNA. So then you have DNA, and then you can do your regular reaction with DNA. And so we measure that in real time, the amplification in real time using this machine that looks like this that's just over in the lab. And if you need a tour, come by my office and I'll take you on a tour of the gear that we have. So we do this every week. We're in the middle of doing it right now. What is it that we're measuring weekly? So weekly, we're measuring female abundance of Culex tarsalis mosquitoes and Culex pipiens mosquitoes. So we report how many mosquitoes were there per trap night. We report the West Nile virus infection rates in mosquitoes. So how many per 1,000 tarsalis and pipiens females have viral RNA. So we're testing pools of 50. Our assumption always is that in any given pool of mosquito, any given pool of mosquitoes, only one of them is infected. I think it's a very solid assumption, but it's an assumption we have to acknowledge. So we report infection rates as positives per 1,000. And then we report the vector index, which is an amalgam of this information on abundance and infection rates, which I'm going to show you right here is how that works. So basically what we do, we calculate the vector index of the sum of the abundance of mosquitoes per trap night times the infection rate across different species. So in our case, it's always just like tarsalis and pipiens. We have how many tarsalis were there times how many were infected, plus how many pipiens were there times how many pipiens were infected. And And then we report that. And here's the sum of what the data looks like. This is from 2014, and we have ID numbers, and this is the epidemiological week of the year, the date that they were trapped, and so forth. And these are all negatives, but then we have positives, and here we see that these are all tarsalis. That's how it usually looks, even towards the end of July. But you're seeing that there are pipiens pools that are coming. This is kind of what our vector index calculation looks at. We divide the city into 4 zones, and there's a reason for that I'll tell you about in a second. But if you say, you know, here's the pipiens number, here's the infection rate. So none of the pipiens are infected. We have 24.5 tarsalis per trap night, and this is how many are infected. So that gives us, this is our vector index. Then we can add this up for the whole city, or average it for the whole city. And that sort of gives us how dangerous the situation is. So the reason that we do this, dividing the city into quadrants, is that it helps people decide at the county level and at the city level whether they're going to go and spray. And so they have thresholds at which they're going to say, all right, this is dangerous enough that we're going to go and spray. So typically in Fort Collins anyway, that's like 0.75 would cause somebody to say, okay, we need to take some type of action. The problem is that West Nile risk is really focally concentrated, even in a place as small as Fort Collins. So it could be, you could have a really high infection rate over by the Fossil Creek Reservoir, but in my neighborhood in Southwest Fort Collins, there's nothing that's infected. And so what you want to be able to do is to say, all right, this part of the city is a problem. We want to do control there. And what that ought to, in theory, allow you to do is to like not dump pesticides all over the town of Fort Collins, which are not necessary, and give people the public health services that they need to protect them while there's still a chance to protect them. The other thing, because what could happen is like you could get to 0.75, but by the time it's 0.75 citywide, the part of the city that actually needed the help, they needed that help like a month ago. Does that make sense? So that's kind of what this is all about, is trying to scale the response to the place where it's needed at the time that it's needed. I'm running out of time, so I'm just going to show you just kind of what 2023 was like. We tested a heck of a lot of pools. And this goes to the question, Caitlin, that you had, I think it was you earlier, like where do you see what's the timing of all these things? And so this is weeks of the year. So this starts like June. We're about in week 24, 25 right now. This is how many vectors were out there. And what's really obvious is that starting basically next week in week 26, we had, you know, about 10 times as many mosquitoes in our traps as we generally have. And so that's like very concerning. And it stayed high and continued to peak at, I mean, like several hundred times the normal Culex abundance. So if you look over here, like you start to see a vector index at week 27 after you had a week of warning already. And then, you know, the vector index peaked at week 29, but it stayed high. The one thing and part of the reason that I said that what I see tarsalis doing is generating this big pool of mosquito of infection, and then other things kind of sustaining it, is this abundance, and this is mainly tarsalis. I would say it's almost all tarsalis. This really high abundance falls off pretty quick, but the vector index does not fall off right away. The vector index sticks around. And so I think that, you know, you see that the abundance drop, but the damage is kind of done, right? So if you wanted to really intervene, you'd want to be intervening kind of back like end of June, beginning of July, to try to knock this down, because once it gets up, it's hard for it to come back down. You know, there's a lot of really, really different reasons that you could kind of dream up for yourself that this is of why this is happening, and why it happened in 2023, but we don't really know. I think there's a lot of hypotheses to explore, but I don't think I'm going to go into them now because there's just 5 minutes left, and I want to save a little bit of time for discussion if there is any. Does that work for you, Tim?
Yeah, that's good.
I can stop screen sharing too.
Yeah, so
What
if
do you
has
have
questions.
about
I have one. How long into kind of the fall and winter do we still keep seeing cases? Like, is it till the first kind of frost and snow, or can they keep surviving that until it gets like really cold?
By cases, do you mean mosquito positives or bird positives or?
Well, I guess more so the mosquitoes. Like, I don't know how like sturdy they are in surviving harsh weather and how long they kind of stick around at either with disease or without.
Yeah. So we stopped testing like early mid-September. So we're not testing after that. But the reason that we're not testing after that is that the mosquito abundance goes way, 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 way down. And so they don't tolerate freezing too well. That's one thing that causes the populations to fall back. But there are other kind of environmental cues there that they're going off of that cause them to do things like get ready for the winter. I don't actually know how Culex tarsalis overwinters, but in the Culex pipiens, Culex quincofasciatus group of mosquitoes, what generally happens is the amount of light causes them to enter, the amount of light during larval development causes them to enter a state called diapause, which is really an ovarian diapause. So what that means is that they become reproductively inactive, and they're storing nutrients and fat in fat bodies. And so then they stop when those mosquitoes emerge, they've basically put all their resources into long-term survival. And so they then focus, on finding a place to overwinter. And so they'll go into things like storm drains that are sheltered from like the bitter winter cold, or they'll go into garages, sheltered, spaces like that. And they just rest and they just hang out all winter long. And then as temperature starts to increase again in the springtime, that's a cue for them to start moving from inside these tunnels out. And so you can start to see them moving kind of as it warms up. So I mean, that's kind of a long-winded answer to not really the question that you asked. The real answer is we don't know because we don't test them that far, but there's just not that many mosquitoes around by the time we're done testing. It's like, here's a few pools to test.
I'm trying to think about ways that people in more like rural communities can try to, um, I don't know, manage their West Nile situation. So I'm wondering if you have any advice for people in more like rural areas, because that's a lot of area to try to like spray. Do you look for like hot spots or?
Yeah, I mean, we don't know very much at all about what's happening with West Nile virus in rural places. Brian Foy, the director of this whole program, has a project that specifically is looking at transmission of West Nile kind of out along the Platte, away from where we are now. And what we're hoping that we're going to learn a lot about it. He also is developing some tools that could help people where there's not going to be spraying and there's not going to be, the city of Fort Collins or Larimer County coming and bailing them out, to kind of take matters into their own hands. But always, like, the best advice is to, don't get bit by mosquitoes, wear the bug spray, wear long pants, do your best to avoid mosquito bites, especially when you have some idea that, well, out here in Alt, we don't know whether there's West Nile virus, but there's West Nile virus over there in Fort Collins, so maybe I better pay a little bit of attention. And like we think about interventions as spraying, but there's all kinds of things people do. In fact, like there's places in Colorado that just categorically will not apply pesticides because it's just against what their values as a community are. They would rather just not do that. That's a fine decision, but then what do you do instead, right? That tends to be things like, putting out flyers, putting out public health notices, hoping that people will listen to them. There's a wide array of actions that could be taken. All right, well, thank you, everybody. Nice talking to you.